Is science racist? Yes, it is. The real question is, what are we going to do about it? Welcome to What's Up in Science with Dr. Tetikol. New paper, just in, a draft human pangenome reference. Whatever that means. We all know what a genome is, right? It's basically the entirety of our DNA with all of its 46 chromosomes. But what is a pangenome? Before we answer this question, we need to clarify one thing that each and every one of you is thinking about right now. How do you prove you are a human? How indeed is a human being defined? Or do we just take it for granted? Well, historically, we have defined humans based on their physical features, such as their body shape, their skeletons, the distribution of hair on their body. But today, of course, we know better to look at the DNA, right? But how does that work? Well, you collect a sample from an individual. It can be blood sample or a tissue sample. You extract the DNA from that sample, and then what do? Well, you compare the DNA you collected from the sample with the reference genomes that we have available today. Yes, today we have a library of reference genomes for many species that is curated and managed by the Genome Reference Consortium. Well, the question you should be asking to yourself right now is, how does one read the DNA? It's not like it's a book that you can just open up and read. Or is it? No, not really. But let's roll with that analogy because I think it's going to prove very useful. If your DNA was indeed a book, it wouldn't be like this, but more like that. What this means is it's not your usual book that you can easily read. So the state-of-the-art approach is to actually put that book through a shredder and get much smaller but readable pieces of DNA. After that, you try to reassemble those small pieces into a traditional book that you can actually read. The problem is the human genome contains 3 billion of letters, TCGA. You remember those from your high school biology class? So after the shredding process, you get not thousands, not millions, but billions of pieces of DNA. So it's a little bit difficult to reassemble all of these pieces into an actual book that you can read. By the way, the process of shredding the DNA into much smaller billions of pieces is called the next generation sequencing. A rather poor choice of words if you ask me, because today we already have the next generation sequencing technologies. But that's a different topic for a different video. So in practice, you take all of these shredded pieces and compare them to every single reference genome we have in the library. So what's the problem here? Well, the problem is these reference genomes that we have in the library, they're not as high quality as we thought they were. More specifically, they're not as representative as we thought they were. You and I are both humans, but we're not the same at the DNA level. Otherwise, we would be the same person. So compared to this human genome reference, our DNAs look different. Maybe yours is very similar to it. Maybe mine is very distinct from that. And that's exactly the problem. It turns out this human genome reference has a predominantly white European background. So if you're African, East Asian, South Asian, Middle Eastern, South American, well, this reference is not going to work as well as it's supposed to on your genome. Just to give you an example, the genomes of African ancestry contain 300 million of nucleotides that are entirely missing from this reference. That's a whopping 10% of the entire reference. 10% of their genomes is missing from the reference. There's a plethora of algorithms and genetic tests that are based on this reference that are not going to work properly for many of us just because of our ancestry. The genetic tests that supposedly tell you might have this or that disease, nope. The tests that predict your physical characteristics, such as your height and weight, think again. The ones that tell you what you're supposed to eat based on your DNA? No way. But there's good news. This paper assembles a new human genome reference. But this time, they're using 47 people all around the world, capturing the genetic variation that we have across all those different populations and ancestries. You might be thinking at this point, 47 is such a small number given that there are more than 8 billion of us in the world today. Well, it's still 47 times better than what we had previously. So this is a great step forward. They improved the detection rate of large genomic variations by a whopping 104%. And we call this new reference the pangenome. The prefix pan comes from Greek. It means all. So in essence, pangenome means all genomes or all-encompassing genome reference that represents all populations around the world. So the next time your doctor wants to run a genetic test, you ask, which reference are you using? I don't want the genome. I want the pangenome. Joking aside, it's going to be a while until we see the fruits of these new developments in practice. But until then, I'll keep you updated with what's up in science. If you like this video, please subscribe and smash that like button. Why? Here's the paper. It says that you're supposed to subscribe to my channel, published in Computers in Human Behavior.